Okay, so here we're going to talk about protein structure and how we build a protein. It covers in AP Biology topics 1.3, 1.4, and 1.5, at least the proteins aspect of those topics. Now, when we talk about proteins, though, before I get started, I really want to stress that the whole job of a protein, no matter what protein it is, its job is dependent on its shape. So in proteins, whether they're enzymes or hormones or pigments, the shape of the protein determines its function and its ability to work. We'll see a few examples of this at the end of the PowerPoint, but as we talk about the structure and how you fold and how you build a protein, it is so critically important to all of life that proteins are folded up correctly. So when we talk about proteins and how we build them, though, it's important to know their building blocks. So we start with monomers of proteins are amino acids. And then the polymer of a protein is a polypeptide. And we'll understand where that word polypeptide comes from in just a few slides. So here we have our amino acids, and we have our chain of amino acids called a polypeptide. Now there are 4 steps to building a protein. You have your primary structure. So while this looks like easy, cute, and pretty with colors, it really is actually a bunch of carbons, hydrogens, oxygens, and nitrogens attached to each other. So when we look at a primary structure, I'm going to model each of these with paper as we go, but the primary structure of an amino acid looks like this. It's a primary structure of a protein is a chain of amino acids, and this primary structure is determined by the order of your genes, of your DNA, your A, T, C, G, what is the sequence in your DNA? Your DNA are the directions of which order to connect these amino acids. Then we have our secondary structure, and then we have our tertiary structure, as well as our quaternary structure. And in this video, we will talk about all 4 of these structures and how they form. Okay, so let's start, though, with looking at amino acids in more detail. So we have 20 different amino acids in life, and the basic structure of an amino acid is the same for all of them. So basically, all amino acids, all 20, have this same part. They all have an amino group, a carboxyl group, and a hydrogen attached to a central carbon. There's also a variable side chain, an R group. And out of our 20 amino acids, it's this R group that is different for each of them. And it's this R group here that actually will determine how a protein folds up. When R groups interact with other R groups, they form bonds or attractions or repulsions, and that ultimately is what changes or determines the shape of the protein. So this part, this R group, is what's different for each amino acid. Okay, so when we look at 2 amino acids, here we go. When we want to build a polypeptide, just like we saw in the last video on topic 1.2 and 1.3, we can actually remove a water on a hydroxyl group. Now, we are removing the hydroxyl group from the carboxyl group. So right here's where we're removing the water from. And then we remove a hydrogen from the amino group. This is how water is formed. And then the 2 amino acids will bond together, forming a peptide bond. This peptide bond is a covalent bond. Now, I want to emphasize, you will never see an amino acid like here, if I have my central carbon, you will never see it between 2 amino groups. You will never see it, it will never form between an amine group and an amine group. It'll also never form between a carboxyl group and a carboxyl group. It's always going to be between a carboxyl group and an amino group. And as you build this polypeptide, as you build this primary structure of a protein, the amino group is always going to be in the front or at the beginning. And then the tail or the terminus, that's a fancy word, the terminus, I don't even know how to write clearly. The terminus end of this polypeptide will always have a carboxyl group. So when we talk about, let me go ahead and just redo this a little bit clear for you one more time because it's so important. when we build a peptide bond between two amino acids, the hydroxyl group of one amino acid and the hydrogen of another amino acid from the amino group are removed and form water. And then when the new covalent bond is built, we call that covalent bond a peptide bond. So you can imagine in a chain, we did dehydration synthesis. So this process here is called dehydration because we removed the water, and synthesis because we built something. So here you have your two amino acids. When you remove a water, you get a peptide bond. So what I was saying is when you have a chain of amino acids, you can see lots of water would be formed. I don't know where I was going with this, I'm sorry. But here you have your amino group, and then you have your carboxyl group. And then in this chain of amino acids, that's what I was saying. So when you have a chain of amino acids, it's called a polypeptide because the many peptide bonds. So in this paper version here, I actually highlighted all the peptide bonds pink. So you can see how it would be called a polypeptide. Now here, if you hit pause, you can try and figure out how many amino acids are pictured. Okay, and there's actually 4 amino acids pictured here. And if you think about it, how many peptide bonds there are? Good, there's 3 peptide bonds. So while enzymes built, or ribosomes built this polypeptide, you would have produced 3 waters along the way. Now, if you were to split and break apart these amino acids, it would require water. So to break this peptide bond, you'd have to add a water to this to split it apart. We call this process hydrolysis. Lysis meaning to split, and hydro meaning water. So here we have hydrolysis. This happens in our stomach. So if you eat some peanut butter or some chicken or something with protein in it, your enzymes in your stomach will actually do hydrolysis and break apart your polypeptides into individual amino acids for you to use, like to build your muscles or your antibodies or enzymes. So then later, when you go to build like new muscle fibers or other proteins, you would actually do dehydration synthesis to build those. Okay, so let's go ahead and talk about the primary structure. So the primary structure, the directions to build it, come from the sequence of nucleotides in your DNA, the A, T, C, G, A, T, C. Your genes in your DNA are what give the directions to how to build the protein. However, DNA is stuck and safe in your nucleus. Therefore, we need to make a copy of that instructions, and we make that copy into RNA. This is why we call the RNA messenger RNA, because they're carrying the message of how to build the protein. And then a ribosome, an organelle, will actually read that RNA and build the protein in the correct order of amino acids. And this is where that genetic code, the code for all life, comes into play. So here in our primary structure of a protein, we have here a polypeptide. This is your chain of amino acids. All of these amino acids are attached together by dehydration synthesis, forming peptide bonds. You would have peptide bonds between each of these amino acids. But how does the cell or how does the ribosome know the order to attach these amino acids? It's from the directions in DNA. So if I looked at an RNA sequence, for example, you would read it in groups of 3, and I know this is another unit and a different topic altogether, but to tie it into your future biology topics, this genetic code is what we use to figure out which amino acids to attach and which order to build them in our primary structure. Okay, so now we need to figure out, once we have built our primary structure, our sequence of amino acids, how does it begin to fold up? Because right now it's just like a straight polypeptide. Well, our secondary structure begins that folding up process. So this is where, from this point on, we're going to talk about different bonds that are involved in having that polypeptide fold up into a 3-dimensional structure. So in the second step of protein folding, we have what's called the secondary structure. And the secondary structure has two possibilities in their form. We have what's called the beta pleated sheet as well as the alpha helix. So when I look at a paper version of this, you have some that are going to be like an accordion shape. And then you have some parts that are more curled or more round, kind of like the helix in DNA. But one polypeptide can have both types of hydrogen bonding of secondary structures. So in one polypeptide, some areas might curl into your alpha helix, some might form into that beta pleated sheet. But really, when we look at this secondary structure, we want to think about what bonds are responsible for this. So as we stare at this picture, we can see that there's hydrogen bonds that are responsible. We can still see, though, the peptide bonds that were formed in the primary structure. You still have your peptide bonds are still there, but now we're starting to fold up, and this is responsible, hydrogen bonds are responsible for this in both the beta pleated sheet as well as the alpha helix. Okay, so the secondary structure is due to hydrogen bondings between like the backbones of the amino acid polypeptide. So here is a three-dimensional animation of, I believe it's hemoglobin, which is the protein that carries oxygen in your blood cells. You can see here there's some areas that have that alpha helix, that spiral shape, and then some that are maybe more like folded. That is going to be your beta pleated sheets. All right, so now we're going to talk about the tertiary structure. And the tertiary structure is really dependent on the interactions of the R groups. So how these R groups interact with each other is what determines how this protein is going to fold. So when we look at the different chemical properties of R groups, you have some side chains that are nonpolar. And remember, nonpolar and polar do not go together. They do not attract each other. Oh my gosh, sorry. And so when we look at our polar side chains, you have some that are polar, and polar are attracted to each other, and polar also interacts well with water. Non-polar side chains are going to be repelled from water. And then we have some side chains that have positive or negative charges, some ionically charged side chains that have positive and negatives, and then those will interact to form hydrogen bonds. Oh my gosh, not ionic bonds, sorry. So let's go ahead and look at this. So here I have a series of amino acids. Now, I can see that all 5 of these amino acids have the same like top parts. They all have your amine group and your carboxyl group. Now, I did pick these, the style of showing amino acids on purpose to show you that sometimes it's not always displayed in that same image from earlier in the video. But what you do notice in blue here are these different R groups. So some R groups will be nonpolar. Now, some of them, though, have a molecule or an atom of sulfur as part of it, and that will be important in folding up of proteins. Then you have some that are going to be polar, as well as negatively charged and positively charged. Okay, so let's see. How do these R group properties really determine how a protein folds? Okay, so when you build the tertiary structure of a protein, this polypeptide chain that we have, the R groups, when you have, you will have some hydrogen bonds begin to form between them, between some of the amino acids, but you also are going to have, when you have the amino acid methionine has a sulfur in it, and the amino acid cysteine has a sulfur in the R groups. So when they come in contact with each other, they're actually, the sulfurs from each one are going to form a covalent bond called a disulfide bridge. So if I had like a sulfur right here on this R group and a sulfur right here, they would actually come together and form a disulfide bridge. So now I have the beginning, like a bond forming. However, if there was a mutation and these sulfurs were not in these locations, you could have a different shape entirely. For example, what if this sulfur R group was still here, but maybe there's a mutation and now the other one is right here. So now you have a totally different shape to start out. So where the R groups matter and the bonds that form between them will determine how this protein will fold. So if you have mutations and you change your DNA and you change your placement of amino acids, those R group tails are in different locations. Well, now how they naturally will form together is going to be different, resulting in a different three-dimensional or 3D shape. So other bonds that happen, though, that influence the tertiary structure, we have our hydrogen bonds, our disulfide bridges, but we also have ionic bonds. So when we had those positive and negatively charged R groups, they will find each other. So here's maybe my sulfur. Maybe this right here is positive and over here is negative. So they're going to pair up together in an ionic bond. So your positive and negative amino acids are going to find each other and they're going to form ionic bonds. And then we have, since you saw earlier in the diagram, that almost half of the amino acids are nonpolar. And a good large amount are also polar. So you have your hydrophobic interactions. Your nonpolar, okay, let's think about this deeply. So proteins live within your cells as well as they could be floating in your blood or outside of your cells. Regardless of their inside or outside, your proteins are surrounded in water. So your polar and your nonpolar amino acids are going to respond to that. If you think about your nonpolar amino acids that repel water, as this protein folds up, you're going to find those nonpolar amino acids towards the center, away from the outside where there's water all around surrounding this protein. So on the outside of your protein, the edges, you're going to find your polar amino acids or your charged amino acids. So your nonpolar amino acids are going to group together in the center, as well as your polar amino acids will be on the surface, and that will influence the overall shape of your protein. So if you have a mutation that switches a polar and nonpolar R groups and you change them, well, now how they used to attract and pair up, now they're repelling, and now that protein's whole shape could be disrupted. So these R groups and their order that they are placed in their primary structure, it definitely matters with how it will ultimately fold up. Now, so here you get your tertiary structure, and this is going to be based on hydrogen bonds, ionic bonds, disulfide bridges, nonpolar amino acids here, polar amino acids there, and it causes this overall 3D shape. Now, that is your tertiary structure. That is the toughest one for students, so I would read the book, I would watch other videos, I would really put my time in understanding how that tertiary structure forms. Then we have the quaternary structure. The quaternary structure is kind of cool. The quaternary structure is basically where you take multiple polypeptides and they join together. So here I have 3 polypeptides coming together to form my quaternary structure. That one is the easiest. Okay, now let's go ahead and talk about, well, what would happen? Oops, I'm sorry. Here's a summary. So you have your primary structure first, followed by the primary structures, the order of the amino acids, held together by peptide bonds, with DNA and RNA giving the directions of how to build them. Then you have your secondary structure, which is based on hydrogen bonds, as well as your tertiary structure that's based on the interactions, the hydrophobic interactions, ionic bonds, disulfide bridges, hydrogen bonds, and then your quaternary structure, which is multiple polypeptides together. Okay, so the last topic here is the relationship between DNA and RNA. So normal genes with normal good directions code for your mRNA, and your mRNA is the directions to build a protein. Now, sometimes though, mutations happen. Actually, mutations happen constantly. Every time your DNA makes a copy, like replicates, is about 30 new mutations. But they're not always in critical, important spots. Anyway, so now if a mutation happens in a gene, in a coding region, that mutation could alter the order of the amino acids, which could result in an abnormal shaped protein. Now, if the amino acids change, it very likely will change the shape of the protein and ultimately its function. Now, sometimes, though, that mutation results in a stop codon and no protein is made. Now, this could be the case with people who are animals who are albino. They just don't make the protein melanin for pigment. Okay, so let's go ahead and see an example of this. So there's different kinds of point mutations, but let's pretend, and I don't have this memorized. So here we have some polar amino acids. So if you have some basic R groups, now it wouldn't be too devastating if the mutation caused lysine to be replaced with arginine. If they're both like the same properties, not a big deal in folding up. However, if the mutation caused lysine to then be replaced with threonine, a polar amino acid, that could have devastating consequences for how that protein folds. So a real life example of this is sickle cell anemia. So hemoglobin is the protein that's made of 4 polypeptides, and this is the protein that carries our oxygen in our cells. Here's your DNA that codes for the order of the amino acids to build this polypeptide. Now, one point mutation, one spot in the protein or in the gene in your DNA has a mutation that changes the amino acid. Now, the R groups on that amino acid are now different. And how that protein folds up, it actually, instead of folding up in a circle, will actually fold up in a straight line. And this is why people with sickle cell anemia have like a stretched out red blood cell. It's because that hemoglobin is causing like a straight chain, which causes the blood cells to like straighten. And this is like a life-threatening mutation, all because of one change in the DNA and one change in an R group causes the entire protein to fold differently. So its structure has changed and therefore its function. So here's how it should look. So you see your 4 polypeptides. You can see them in their tertiary structures joining to form the quaternary structure. However, in sickle cell anemia, that is not the case, and now you have problems with how your blood flows. You can see these straight chains right here. Okay, and then our last example, I just wanted to touch base on another example of a protein not folding correctly. This is just a picture of the cell membrane, but we have important protein channels that like allow ions and molecules to move in and out of our cells. And it's important that our proteins fold up correctly to fit into our cell membranes. However, the disease cystic fibrosis is the result of a mutation in one of these proteins where it doesn't fold correctly, and now the ions cannot travel through, and it causes all kinds of disruptions in the lungs and the respiratory systems. And that's all due to just a misfolding of that protein. It has different amino acids, so therefore the structure is different, and now the function no longer works. And it has devastating consequences for people with cystic fibrosis. Okay, and that is my lesson on protein structure.